let's look at the spinal cord. The spinal cord is sitting within the vertebral column. And the vertebral column has different types of vertebrae. You can notice that the vertebrae up here look different from the vertebrae here. Do you see that these have very long processes and these don't? And then down here, the processes look different. We're not going to concern ourselves with the different types of vertebral bones, but these are cervical, these are thoracic, and then we get into lumbar and sacral vertebral segments. And sitting inside this vertebral column, if we turn it around, inside here is the spinal cord. This is where the spinal cord has to sit, and it's a small area, but there's a little bit of room. It's unlike the cranium where there's no room. The brain is right up against the dura is right up against the skull. In this situation, there's space between the dura and the side of the vertebral column. So the spinal cord is inside of this column, but what happens during development is that the spinal cord does not grow as much as the vertebral column does. So as a result, in the adult, the spinal cord ends at about right here. It ends about right there, which is on me, it's just a little bit below your rib cage. So it's ending right about here. That's the end of the spinal cord. But there is an anatomical rule, which is that every segment has to leave the spinal cord to go out and be a nerve through the appropriate foramen, or hole. This is the hole. This is where nerves come out of. And the first cranial, or the second cranial nerve, the second cervical nerve, so these are cervical, thoracic, lumbar, sacral, and each nerve has to come out from the appropriately named bone. So cervical 3 has to come out by the third cervical vertebral. The thoracic 10 has to come out by the 10th thoracic vertebral bone. So because the spinal cord is so much shorter than the vertebral column, what happens is that these nerves have to travel down before they reach the place where they're going to exit the vertebral column. So we have a spinal cord, but by here the spinal cord ends, but there are still nerves that are coming down to exit. And that causes a situation where we have, at the end of the spinal cord, we have all of these roots. These are roots that are on their way to exiting below. So in this case, here's the end of the spinal cord. This is the very end of the spinal cord. But by this point, there are all these nerve roots that are coming down to exit farther below. Let's take a look at another one. So here we can sort of move aside these roots, and we come to the end of the spinal cord right here. That's called the conus medullaris. It's the end of the spinal cord, and below it are just roots. This is called a horse's tail, or cauda equina. So it's called the cauda equina, and this cauda equina is down here. It's sitting in the vertebral column down here. So what's the importance of that? Well, the importance is that if you wanted to put a needle in somebody to try and either test them to see if they have an infection in their cerebral spinal fluid, or to administer drugs to them, well, if you do it up here, you risk impaling the spinal cord, and that's going to be disastrous. But if you do it down here by the cauda equina, as you put the needle in, imagine that you put the needle in, well, the nerves just kind of move aside. So if you put a needle in here, you're not going to actually puncture a nerve. They'll move aside. So this is a relatively, it's a relatively low risk, not without risk, but it's a relatively low risk procedure compared to if you went up into the vertebral column at a place where you could actually puncture the spinal cord. That would actually never happen. So that is the source of what's called the lumbar puncture, but which is known, I think, in common parlance as a spinal tap. So this is a spinal tap. You're doing the spinal tap down here where there's no spinal cord, but there's all of these roots, the cauda equina. So let's look at one more thing. What's probably the most common type of pain that we have? Well, it would be I think it would be a close call between a headache and a backache. They're both pretty common, but backaches are really common. What are backaches? What happens? Well, when the nerve comes out, it comes out from this opening right here. And this opening in this individual is pretty big. But you can see in this individual, there's much less of an opening. So look here, there's much less of an opening. Why? Because the discs, these things right here, these discs have compressed and there's less space between them. And so now there's less of an opening. The nerve can get pinched there, it can get squeezed, it can get otherwise tickled, and that is not going to feel good, okay? And so this is one of the big reasons for back problems is that these intervertebral foramens, these holes from which the nerves exit, get small as we age compared to what they hopefully are normally. There are many other reasons for back pain, but that is one that we can look at on a skeleton. Okay, from a whole brain, it's just so hard to see the parts of the brain because the telencephalon, the cerebral cortex, is really hogging the view. All we really can see is the cerebral cortex. So what we're going to do is look at brains that are cut down the middle. This is the mid-sagittal cut, and we're going to look at the medial surface. And when we do that, here's mid-sagittally cut left half of the brain. You can see, we're just going to remind ourselves of some things that we've already seen, just because I think a little bit of repetition helps things sink in. So this membrane here, this is the arachnoid. This is the left cerebral hemisphere. Down here we have the cerebellum. Between the cerebral hemisphere and the cerebellum, right here there's this space, and what's in that space? The tentorium, that fold of dura. It sits right there. So now let's look. The foramen magnum is going to sit like that. The foramen magnum is going to sit like that. So this is spinal cord, and this is now brain. The whole thing is brain. This piece, as we go around like this, oops, here, like that is all hindbrain. And there are three parts to the hindbrain. There's the cerebellum, there's the pons, and there's the medulla. In front of the pons, and then in front of the hindbrain is this structure right here, which is the midbrain. Now this brain was cut just off center, and I know that for a few reasons. One reason I know it is because there's a channel right here, and that channel is very narrow, and it's right on the midline. And so my probe is in that channel, but the cut is not there. So we're going to move over to this. This is actually from a different brain. And you see here, once again, the medulla, the pons, the cerebellum, and this piece right here is midbrain. And the midbrain is marked by this narrow little channel called an aqueduct. Okay, so that's an aqueduct, the cerebral aqueduct. This is the midbrain. Remember, we talked about the two colliculi, the superior colliculus and the inferior colliculus. The superior colliculus is important for orienting movements. If a bird flies by, you look at it. If somebody taps you on your shoulder and you look at them, you use your superior colliculus. And as a result of this superior colliculus, you can have something called blind sight, where you can follow moving objects. Your eyes will follow moving objects, but you cannot actually perceive them, because the information doesn't get to your neocortex, no perception occurs, but you're able to follow them using your brainstem, using your midbrain superior colliculus. Okay, in front of the midbrain is this area, which is the thalamus. And the thalamus is this area right here. It's got there's a little divide here. The bottom half of the thalamus is, or the bottom half of the diencephalon is the hypothalamus, and then up here is the thalamus. This structure right here, this is the corpus callosum. The highway that is connecting the right hemisphere to the left hemisphere. So through here, there are neurons that start, say, right here in the right hemisphere. They go down through the corpus callosum, and they end up in the analogous place in the left hemisphere. So it's just joining the two hemispheres. This is cut in cases of generalized seizures, where a person has a seizure that starts on one side of the body and goes to the other side of the body. If these don't respond to drugs, and if they're otherwise intractable, then sometimes the surgeon will come in here and cut the corpus callosum. Remarkably and interestingly, you actually have to do fancy tests to actually see that there's a problem, that the corpus callosum has been cut. Normally, you would never know it if you met somebody with a cut corpus callosum. Okay, so let's just go over the areas again. This is all telencephalon. Down here is diencephalon. This piece right here is midbrain. Here is pons, cerebellum, and medulla. These three make up the hindbrain, and here is the spinal cord. So here, what we've said is that the telencephalon expands to cover over the rest of the brain. And in fact, what we also said is that the telencephalon is joined to the rest of the brain only by a couple of places. And I want to show you that. I showed you that this is the top of the diencephalon here, and it's separate. But what I've done in this dissection is I took it out. So now what we're looking at is I've taken out the telencephalon. I've taken off the telencephalon. We'll look at that in a moment. I've also taken off the cerebellum. So what we're looking at is the back side, this side, the back side of the medulla, the pons, and the midbrain. And now here, this is the diencephalon. This is the thalamus. If we look around on this side, now we see it looks a little bit more familiar. Here's the medulla, here's the pons. We can see a little bit of midbrain. The midbrain's pretty small. And then here is the base of the diencephalon, which is the hypothalamus. So let's look at what we removed. What we removed is the telencephalic cap. So here's the front, here's the back, this is the middle, and you can see that most of the brain is the telencephalic cap. And the rest of it, the brainstem and the diencephalon just fit nicely into here. And actually most of the rest of it, so this is fitting in like this, exactly like that, and then the cerebellum is right here. And we can take that out. And what you're looking at down here, this piece right here, that is the corpus callosum. That's that fiber tract, that highway that is connecting the two sides of the brain. So now what we're going to do, I realize that I made this dissection and it's not as compelling since I've already done it. We're just going to try and do the same thing on this half of the brain. We're going to try and separate out the brainstem and diencephalon from the overriding telencephalon. And what you'll see is that it's almost all separate. It's joined off on the side, and that's what I'm going to cut. All right, so I've cut that side bit, and now I'm taking this out, and you can see that that's a smooth surface, and now I have to cut one little place here at the front end. I didn't do a great job, but what you can see is that the only cut I ever had to make was the off here on the side. Here we have the pons, the medulla, the cerebellum, pons, medulla, cerebellum, here's spinal cord. And the only thing that joined this whole part of the brain to the telencephalon is off here on the side. Let's look at it again. You can see this smooth surface. If I'd done it a little bit better, I would have gotten more of this smooth surface. This is the top of the diencephalon, and the telencephalon just sits on top of it. It is not connected. So we've got this tube and then something coming back on top of it. So there are two areas of the brain where we have what's called a cortex. And a cortex, there's a definition of a cortex, but basically what it is an outer rind, an area that is layered and contains a very particular type of cell, and it sits only on the outer surface of a brain structure. And the two areas that we have cortices, one is the telencephalon, the cerebral cortex, and the other one is in the cerebellum, the cerebellar cortex. So what's similar between these two areas? Well, they both have these hills and valleys. They're named different things, but they account for the same thing, which is that they increase the surface area, because cortex is only there where there's a surface. So to get more cortex, you have to make more surface area. In the cerebral cortex, that's done by making these hills and valleys that are called sulci and gyri. And what you can see, I've taken a piece of brain and I've cut through it, and what you see here is gray. Do you see this gray that just decorates the outer surface of the brain. It's the rind, that's it. It's not very thick. That's the cerebral cortex, and underneath it is white. Why is this gray? Because there's not very much myelin. Why is this white? Because there's a lot of myelin, because there's a lot of axons. So gray matter is rich in cells, and white matter is rich in axons. This gray matter that forms the outer surface of the cerebrum is the cerebral cortex. When we look at the cerebellum, we see the same deal. It's just smaller. The hills and valleys are much smaller. In fact, they're called folia. But the name of it is not that important. What's important is that you can see that there's this outer gray area served by a inner white matter. Okay? So this is the cerebellar cortex, and this is the cerebral cortex. This type of structure is what's contained everywhere you see this. All over this telencephalon, that's all cortex. So that is the final way that we've expanded the surface area of the cerebral cortex. But let's remember back to how the central nervous system and how in particular the brain develops. It develops from a hindbrain swelling, a midbrain swelling, and then a forebrain swelling. And the front of that neural tube is called the lamina terminalis. And the front of that tube is right here. It's right there. Okay? So we have hindbrain, midbrain, and forebrain, and it ends right there. That's where the tube ended. So what the heck is all this? Well, all of this is the brain, is the telencephalon saying, I want more space, and growing back over the rest of the brain. So, in fact, as we go like this, we can see that it's grown on top of the hindbrain. There's a space there. There's no brain here. It's just come back on top of it. And there is a space. Here's the lamina terminalis. It's right in front of the optic chiasm. This is the front, this is the back, here's the cerebellar cortex, here's the cerebral cortex, this is arachnoid. And what you can see is that the cerebrum actually sits on top of this structure, and it's actually separate, they're not joined. This structure right here is thalamus. That's the thalamus, and it's not joined. There's a space between the top of the thalamus and this overgrown telencephalon that has grown back on top of it. In that space is this small little gland. It actually attaches right there, and here's half of the gland. It's been cut in half. On this brain, here's the gland. Here's the gland right there. It attaches right there. That gland is the pineal gland. The pineal gland is important in circadian rhythm. It secretes melatonin. For our purposes, this gland is pretty important because these pineal cells can become cancerous. They can start to multiply. And if they multiply, they're going to take up more and more space. And you can see that even though this is not part of the brain, it's an actual gland that's got stuck inside of the cranium, that if this expands, we're going to have brain problems because it's going to put pressure. It puts pressure primarily on the midbrain. And one of the things that it does is that it closes this cerebral aqueduct. So can you see that if this were to increase in size, it would press down and close off the cerebral aqueduct? That's a problem, and it causes a condition called hydrocephalus. So the important point here is just to understand that the cerebral cortex, the telencephalon, has expanded so much that it's covering the underlying diencephalon, it's covering the underlying midbrain, and it's covering most of the hindbrain as well. The telencephalon is a hungry beast, and it wants to expand even more, and the final expansion or another expansion that occurs is this temporalization. And so the telencephalon has expanded back here, and then around and forward like a ram's horn. So it's coming around and forward to form this temporal lobe. This temporal lobe is only present in some mammals. It's present in us, it's present in several, it's present in the apes, it's present in several other primates, but not too many other mammals. So this is houses a further expansion of the cerebral cortex over what's present in a general mammal. So just to give you an idea of the anatomy of the cerebellum, let's just take a look at the cerebellum, which I've taken off of the rest of the brain. It's so overwhelmed by telencephalon. So here we're looking from the side, we're looking actually from the right side. Here's spinal cord. This is medulla. We're looking at this from the side, from the right side. Here's the spinal cord. Here's the medulla. Here's the pons and the midbrain. This is the cerebellum. And what you notice here, when it's shown like this, is this big wraparound. That is called the peduncle. That is attaching the cerebellum to the pons. And the pons is the only thing that the cerebellum is directly attached to. The peduncle means, I think peduncle means strap, like a pack sack, and it's strapping the cerebellum onto the brainstem. So let's take a look now. This might look a little bit more familiar. Now we're looking at the mid-sagittal view. We're looking at the medial surface. So once again, this is the back. Here's the spinal cord. Here's the medulla, and here's the pons. And up here is the midbrain. This is the cerebellum. So you see this capsule here, this round part at the base of the pons. Well, it's called the base of the pons. And the base of the pons and the cerebellum are married. In animals that have a small base of the pons, there's a small cerebellum. In animals with a big base of the pons, there's a big cerebellum. This and this evolved together. This is the interpreter. This is like thalamus is to cerebral cortex. The base of the pons is to the cerebellar cortex. Things that go into cerebellar cortex are interpreted by the base of the pons. Now when we look at the cerebellum, and now what I've done is I've cut the peduncle. And so this is the bottom of the cerebellum. The brainstem would be here. This happens to be the front. This happens to be the back. It would be a little hard for you to know that. So when we look at the cerebellum, now we're looking at it from the back. And I spread this. You can see that there's, between these very large lobes, there's a central portion, and that central portion is called the vermis. Let's take another view of that. In this brain, it's a little bit easier to see. So this central portion right here is the vermis. And this vermis is responsible for motor learning and coordination of midline movements, such as walking and gait, balance, speech, and it contributes to eye movements as well. Right next to the vermis is a very indistinct area called the paravermis, meaning next to the vermis. There's no real strict boundary here on the lateral side, but this paravermis area is involved in coordinating and motor learning for reaching movements. And then the lateral lobes are more mysterious right now, but they definitely contribute to hand-eye coordination, to eye movements, and to learning new complicated movements. So this is the cerebellum. And finally, let's look at the cut peduncles. Here are the cut peduncles. This is what attaches the cerebellum onto the brainstem. If it weren't for these peduncles, it wouldn't be attached. Another view of these peduncles is shown here. What we've done in this is we've made a cut like that. We've made a cut. It's as though I took the knife and I cut like that. And what you see is this space here, called the fourth ventricle, is right there. This space here is right here. This base of the pons is right here. And the cerebellum is here. The peduncle is this expanse. All of that is the peduncle. That's the thing that attaches the cerebellum and the pons. The remarkable thing about the peduncles is that virtually all the information is going into the cerebellum and a very little bit of information is coming out. It's 40-1. So the cerebellum is making decisions and it's sending its decision out through the output, but the output is greatly outnumbered by the information that it considers in order to make its decisive output.